Recording in progress.
10 seconds. I'll count you in. 5, 4, 3, 2, 1. Go.
This is Wynn Arias welcoming you to the 9th session of the 21st year of the NIH course called Demystifying Medicine. This is really a course in bridge building. For the most part, it bridges the exciting developments in biology and engineering with medicine, particularly with disease. But the format varies considerably. And as I'll explain shortly, today's topic is one of the more diverse that we have considered. All the sessions are virtual, and it goes worldwide, global. They're on Tuesday afternoons from 4 to 6 Eastern Standard Time from January 4th to May 3rd. And they are accessed through the videocast nih.gov link. The entire schedule for the course is also available using the link shown on the slide here. For those who attend at least 50% of the sessions and pass a not complicated final exam, they will receive a certificate. Today's lecture, it does not have a CME activity, and so there's no need to register for it. Note that all the previous sessions for the past 20 years are archived and available at the link shown at the bottom of the slide. Now, I encourage you to submit questions during the presentations, and that can be done on the videocast display where it is indicated "Send live feedback." These questions will be received, and at the end of each of both speakers today, we will have a question and answer period. The next slide, please. Now, as I mentioned, this is a course in bridge building, and the logo, which at least for those on this side of the Atlantic, is often considered the most famous bridge in the world, is my grandfather's photograph of the construction of the Brooklyn Bridge. And normally the two individuals on the bridge come from different sides of the river, the East River, and are discussing issues of major importance, each representing a different point of view and a different expertise. It's an exercise in communication. Today's bridge is probably the longest, certainly the longest and most extensive bridge, and probably the most important that we could ever consider. And that's the origin of life. And so today's speakers will address this. One is an evolutionary biochemist, and the other a cell biologist. Next slide, please. In reading material for this particular topic, I came upon this quotation from Sidney Brenner. I think one of the things about creativity is not to be afraid of saying the wrong thing. And to a certain extent, that will influence the entire presentation today. Now, this topic really begins about 4 billion years ago, when life arose on Earth from non-living matter. But how, where, and why are mysteries. The earliest life forms were simple and microscopic, yet they had two properties that set them apart from other organic chemical chains in the environment. And these properties are, they could metabolize and they could replicate. This is the origin of life, which will be discussed in the first talk by Dr. Nick Lane, who's a professor of evolutionary biochemistry in the Division of Biosciences Next slide, please. In the Division of Biosciences at the University College London. Nick's research is directed toward a biochemical explanation for the origin of life and the emergence of the eukaryotic cell. The general focus of his work has been on the role of chemiosmosis, that is the generation of ATP through proton gradients across membranes. Nick is an outstanding teacher. Could we go back, please? Is an outstanding teacher and is an award-winning author of 4 books. The most recent, published in 2015, is an extraordinary book called The Vital Question: Why is Life the Way It Is? A new book will be coming out in July entitled Transformer: The Deep Chemistry of Life and Death. And so Nick will be the first speaker who will talk about the origin of life in an evolutionary sense. Now, about 2 billion years ago, life exploded with a dazzling variety. The next slide, please. With a variety and complexity to include now multicellular life in the form of the eukaryotic cell. Well, many things happened between abiogenesis and the rise of the first eukaryotic ancestor. And this will be the subject of Dr. Jennifer Lippincott-Schwartz's presentation. Jennifer, you know, is a senior group leader at the Howard Hughes Medical Institute at Genelia Farms campus. And formerly, from 1993 to 2015, she was chief of the NIH National Institute of Child Health and Development section on organelle biology. Jennifer's approach has been totally dynamic in terms of science and personality. She does research on the eukaryotic cell organelles as dynamic structures. And in part, this is because she has been an innovator in live, subcellular, and super resolution microscopy using photoactivatable organelle-specific probes developed at NIH with the late George Patterson. Jennifer has received numerous national and international awards, including election to the National Academy of Science, the National Academy of Medicine, and the National Academy of Arts and Sciences. And she will be discussing the emergence of the eukaryotic cell. So join us in a $4 billion journey today as we probe theories as to how life originated and how it became more complex. So, Professor Lane, would you please begin?
Thank you very much for the very kind introduction and for the huge honor of being here at all. It really is an immense honor and pleasure, and I'm really looking forward to speaking to you. So I'm going to talk about energy and matter at the origin of life. And these are, I should say up front, ideas that I've been conjuring with for a long time, but lots of people would not agree with me, and you should be aware of that up front. So there's I have no conflicts of interest. This was for the CME program, which apparently is not going ahead. So your learning objective, you no longer need to understand the origin of life, which I'm sure some of you will be pleased to hear, but I will do my best to make it intelligible anyway. The first problem that we all have as soon as we talk about the origin of life is to wrestle with some definition of what on earth is life anyway. and there isn't a good definition of it. I mean, there are actually scores of bad definitions of it, and I think part of the problem, as some wag said, life is not a noun, it's a verb. It's a process over time, it's an activity, and really a better question is what is living. A lot of people, I hope, might have read this famous book by Erwin Schrödinger entitled What is Life, going back to 1944. If you haven't read it, it is well worth reading because it's a beautiful book dating back to before the time that anybody realized that genes were made of DNA. Certainly it was not established in the field in the early 1940s. And Schrödinger talked in quite modern terms about genes and information. This is apparently the first use of the word code script in biology. So during the Second World War and at a time when cryptography was very much, and talked about life feeding on negative entropy. And here his language, to me at least, feels quite antiquated. He says life maintains itself at a fairly high level of orderliness through continually sucking orderliness from its environment. And I have to say, I have no idea what he actually had in mind when he wrote that. He did have a footnote in the second edition where he said, if I'd been catering for physicists alone, I should have let the discussion turn on free energy instead. So just to make sure we're all on the same page, he didn't talk about free energy because he thought it was a more complex term than entropy. I think most biologists would probably see it the other way around. Free energy is really simply, as I see it, the energy available to power work, whether it be muscle contraction or the conformational changes in molecular machines and proteins and so on. And our energy is coming ultimately from burning food in oxygen through the process of cell respiration to generate ATP. And I'm sure that here I'm talking the language of everybody on this call. And of course, we turn over something approximating to our own body weight in ATP every day. It's very difficult to convey just how much energy it costs just to stay alive. And if we start to think about the origin of life, then we have to think about universals across life. And we all know about DNA and the universal genetic code and so on. But it's perhaps less obvious and that there is also a universal form of free energy conservation across all living things. Most people would say ATP, but it goes beyond ATP. It's also the mechanism in which our mitochondria and cells in general, bacteria and archaea and so on, are powering the synthesis of ATP. It's through the use of ion gradients and especially proton gradients across membranes. And that's really a very simple difference in the concentration and therefore the charge between one side of a membrane and the other side of the membrane. And the use of these gradients is as universal as the genetic code itself across all of life. But the machinery that's powering this is extraordinarily complex. And I'm just going to give you a snapshot. I'm sure many of you are more familiar with these kind of molecular machines than I am because I don't specifically work on these, but they are absolutely glorious, marvelous. From the point of view of an evolutionary biologist or a biochemist wondering about how such machinery arose at the origin of life, dumbfounding, really. They're plainly a product of genes and natural selection. And so they seem to have nothing whatsoever to do with prebiotic chemistry. These are the pinnacles of evolution, not the beginnings of evolution. And for that reason, most people have put it aside and don't worry about it and tend to dismiss membrane bioenergetics and the use of this machinery as being something that arose much later in evolution. And we have to look for simpler things at the origin of life. And there's plenty of ideas out there. Reactive gases, cyanide or cyanacetylene, formamide, and so on. Energizing light, UV radiation, lightning, all kinds of dynamic environments, wet-dry cycles in terrestrial geothermal systems and so on. This is the dominant area of this field now. And this has been going for 50 or 60 years, going back really to the Miller-Urey experiment with bolts of lightning to try and effectively energize the gases in those early atmospheres. And it's worked extremely well, and the chemists think they've solved the problem. But from a biologist's point of view, it's, to my mind, this is Frankenstein chemistry. It looks nothing like the chemistry of life as I understand it. And so I suppose at the very least, even if this chemistry is in fact all the right chemistry and life really did start in this way, we still have a gap. We still have to explain, okay, so we've got all of these chemicals. How did metabolism then start? How did genes invent metabolism, metabolism as we know it in cells today? Because this is nothing like the metabolism of cells as we know them. So from a biologist's point of view, I am very uncomfortable with those answers. And I turn to people like Christian de Duve, who try to use biology rather than chemistry as a guide to life's origins. And there's a lovely quote from de Duve here, which I'm going to read out to you. He said, "How did proto-metabolism," by which he meant the geological chemistry, the geochemistry happening in some environmental context, how did that kind of dynamic chemistry, proto-metabolism, come to be replaced by metabolism as we know it? And he says the obvious answer to this question is that the appearance of catalysts, whether ribozymes or protein enzymes or both, was responsible for the transition. We have to ask how catalysts with appropriate properties came to appear. Why do they catalyze the right reactions? And he says the only scientifically plausible explanation is that the catalysts arose through selection. And then the killer line, he says enzymes are selected only if they fit into proto-metabolism. Now, this is hardly an irrefutable argument, but it's certainly a powerful case. And it's basically arguing that metabolism amplified the geochemical pathways that preceded it. And that makes a lot of sense to me. The only problem is that they never really seem to work. People have tried these experiments, and until recently, they haven't worked well. But recently, things have really changed, and I'm going to tell you more about that in this talk. But what kind of chemistry do we actually need to talk about? To do that, we need to try to get back to the roots of the tree of life. And one of the most brilliant scientists working in this field for many years now, over several decades, is Bill Martin. Now, he has some very radical ideas. A lot of people find his ideas very uncomfortable. I certainly did myself. But the longer I've lived with them, the more I think he's bang on. Now, this tree that I'm showing you here is a tree from one of Bill's papers from more than 20 years ago now, and it's basically a conceptual tree. And what he's showing is the eukaryotes up here, which Jennifer will talk about in a while. And the eukaryotes, you can see, are deriving from an endosymbiosis giving rise to the chloroplasts in plants here, and an endosymbiosis deeper down in the tree between a bacterium of some sort that became the mitochondria and an archaeal host cell. And so the point for my talk is that eukaryotes are a derived domain. They don't speak directly to what was happening right down at the base of the tree down here. Now, again, not everybody agrees with that statement, but probably that would be a consensus in this field now. So Luca is the last universal common ancestor of all of life, and it's the ancestor of the bacteria and the archaea, which look a lot alike in terms of their gross overall morphology. And Bill has Luca down here in a hydrothermal vent with two separate emergences from this vent, which is in itself a very radical idea. He's not saying they originated independently. He's saying that they shared a common ancestor that lived in a rocky hydrothermal system. Why would he say such a thing? Essentially because it's plain that life shares a common ancestor, and that's because life shares the universal genetic code, the structure of the protein factories, the protein making factories, the ribosomes, the genes, and therefore the enzymes involved in transcription and translation, quite a lot of intermediary metabolism like the Krebs cycle, and most importantly from this lecture's point of view, membrane bioenergetics. But what's different is what's really shocking here. The cell membrane is different in its chemistry. Very different in its chemistry in bacteria and archaea, along with all the lipid synthesis enzymes that are required to generate these membranes, along with the cell wall. So basically the barrier between the inside of a cell and the outside of the cell are not homologous in bacteria and archaea. They appear to have emerged independently, and that leaves you conjuring with what on earth did the common ancestor have then? It almost certainly had a cell membrane. I don't know if it had a cell wall or not, but it was not like a modern bacterial or a modern archaeal cell membrane. Pathways like glycolysis, the genes involved are not conserved in bacteria and archaea. Even DNA replication, the genes involved are not conserved in bacteria and archaea, only one or two of them. And various proteins that can move, or genes that can move around by lateral gene transfer, including those for encoding the respiratory chain in respiration, don't really appear to be deeply conserved. So this leaves you wondering what on earth was LUCA, this last universal common ancestor. What kind of a cell was it? It's quite a paradox. And the paradox as I see it is membrane bioenergetics are universal across all of life, but the membranes themselves are not. And this paradox could be resolved if life originated in an environment where there were geological proton gradients across barriers that are not necessarily like modern membranes. Is there such a place? Yes, they were discovered 20 years ago by Deb Kelly. She was captain of the Alvin submersible. In the year 2000, she was actually aiming for the mid-Atlantic ridge, to the black smokers at the mid-Atlantic ridge, and discovered about 15 miles away from there this completely different type of hydrothermal system. They're now known as alkaline hydrothermal vents, and Lost City was the one that she discovered. And it's not like the black smokers. So she's holding a lump here of a black smoker, and you can see it's a chimney. And the black smoke would belch out through this chimney. This is a lump of the Lost City, and it looks much more like a mineralized sponge. And the hydrothermal fluids will percolate through that sponge. They don't kind of belch out of the top. So this is a very different kind of hydrothermal system. And instead of being formed by direct interactions between ocean water and magma chambers from the thousands of degrees, the magma. This is water which is percolating down. Let me just put my pointer on. So water will percolate down into the sea floor, 5 or 6 kilometers depths below the sea floor, and react with minerals like olivine, which are rich in ferrous iron and magnesium, oxidizing the iron from ferrous to ferric iron, and that produces warm. In geological terms, warm means 50 to 100 degrees, for example, and strongly alkaline, pH 10 or 11, and saturated in hydrogen gas, bubbling in hydrogen gas. This is kind of caustic soda bubbling with hydrogen gas. They're buoyant fluids. They bubble back up to the bottom of the ocean, and there they react with the ocean waters and precipitate these hydrothermal systems. And they're found not only on Earth, but we know these plumes, for example, on Enceladus. These are alkaline. They're hydrogen rich. They contain other organics and gases like methane. And it seems almost certain now that beneath the icy crust of Enceladus is a liquid ocean, and beneath that liquid ocean, there is hydrothermal systems like these ones on Earth that I'm talking about, that I think were the environment where life started. We find them as well, probably on Europa, and any traces of methane found on Mars, if they really do exist, are almost certainly these same kind of serpentinization reactions between traces of water and minerals like olivine on Mars. So these are processes that should happen on any wet, rocky planet or moon, and they're common even in our own solar system, and should happen probably on tens of billions of planets in the Milky Way alone. The idea that life might have started in these environments was first put forward by Mike Russell back in the early 1990s. And he has been pioneering these ideas. I don't always agree with what he has to say, but he's had some absolutely stupendous ideas. And I'm whittling this down to his key points, really. He sees these vents as electrochemical flow reactors, where we have fluids which are percolating through these this labyrinth of micropores with catalytic walls. The catalysts in these walls in the early oceans, when there was no oxygen and lots of iron around, were probably iron sulfide minerals. There would have been plenty of other things in there as well. So we have alkaline fluids, warm alkaline fluids, bubbling in hydrogen gas, interacting with these catalytic walls, and ocean waters as well, percolating in, full of CO2, saturated in CO2. And in some way, this electrochemical flow reactor is going to drive the origin of life. That's what I'm going to try and tell you in the rest of this talk. So these vent pores are analogous in their topology to cells. And this is my simple attempt to convey what a bacterial cell looks like at its most elementary level. It's basically got a membrane pump here surrounding them in the membrane. Is pumping protons out. And so we have a proton gradient across this membrane. It's relatively alkaline inside, relatively acidic outside. And that's, of course, putting a charge on this membrane because the protons are moving out. And here in the vents, we have an inorganic barrier, thick in comparison. We have acidic ocean waters percolating into this system, and we have alkaline fluids. So we have a similar pH difference, similar magnitude of probably several pH units, and it should also be similar in its polarity. It's effectively acid outside, alkaline inside. Is this just imagination? Am I seeing things here? Or can we come up with a way of going from this simple inorganic system to the way that cells work? I'm actually following here in the footsteps of Peter Mitchell himself, who did wonder and work a little bit on the origin of life. So Mitchell's ideas, he was the person who came up with these ideas of chemiosmotic coupling and membrane bioenergetics. They didn't go over well at the time. There's a lovely quote from Leslie Orgel, who said, "Not since Darwin has biology come up with an idea as counterintuitive as those of, say, Einstein, Heisenberg, or Schrödinger." And it was really Jennifer Moyle who did the pioneering experiments. Mitchell himself was quite cack-handed in the lab, and it was Jennifer Moyle who did the experiments that demonstrated initially to a very skeptical world that there was something in these ideas. Now, Mitchell himself was quite philosophically minded, and here's a quote which I think is beautiful in its application to the origin of life. He says, "I cannot consider the organism," and here he's thinking about bacteria, "without its environment. From a formal point of view, the two may be regarded as equivalent phases between which dynamic contact is maintained by the membranes that separate and link them." So he's thinking of the inside of a cell and the outside world as equivalent phases, with a membrane which is 5 millionths of a millimeter thick, separating these two different phases. Now, the origin of life, there isn't such a thing yet as a cell or as life or as living things. These two phases with a barrier separating them makes much more sense when we're thinking about the origin of life. We're beginning to see how a system can in itself be compartmentalized. I can't work out why I'm not moving forward here. So Mitchell was asking a very simple question, which was basically, how do bacteria keep these two phases separate? How do they keep the inside different from the outside at its simplest possible level? And he recognized that while they have to find particular molecules, protons in this case, recognize them and physically pump them out, and that's going to cost energy. And conversely, it will you'll be able to tap that energy. If you can conserve that energy as they allow them to flow back in again, you'll be able to power work. And mostly when we think about that work, we're thinking about ATP synthesis. But it's not only ATP synthesis. In bacteria, very often the proton gradients are driving CO2 fixation. And that, to me, is the key to thinking about this. So the most ancient cells that we know about grew from hydrogen and CO2. And they're fixing CO2, and to do that, they're fully dependent on chemiosmotic coupling. They require these proton gradients across membranes. So these cells are methanogens, or archaea, acetogens are bacteria, and this is the only pathway which is found in both bacteria and archaea of CO2 fixation. It's called the acetyl-CoA pathway, and it's basically a reaction between CO2 and hydrogen. So these are the gases that we have in these vents. And when they react together, it's exothermic overall, in the case of methanogens, to produce methane as a waste product, and in the case of acetogens, to produce acetate or vinegar as a waste product. So it's overall exogonic, but they don't react very easily. They need a proton gradient across the membrane to drive those first few steps. And what they're doing is using a membrane protein, in this case, the energy converting hydrogenase, which is an iron sulfur protein, to reduce ferredoxin, which is another iron sulfur protein, which then reduces CO2 to make organic molecules. So it's taking the electrons from hydrogen, it's cobbling them onto CO2 to make organic molecules. That doesn't happen spontaneously, and so it's using a proton gradient through these iron sulfur membranes to drive that. And this slightly more complex diagram here, I won't try and explain it to you, but it's essentially saying that first step is electrochemically not favored. There's a difference of about 200 millivolts that need to be overcome to drive that reaction. When you have got through that step, we get into the core of biochemistry as we know it almost immediately. So don't worry about the details on this slide. What I'd like you to notice is this hydrogen is in brown, and from starting with CO2 in this corner, we've got hydrogen, hydrogen, hydrogen through to a methyl group, more hydrogen to make carbon monoxide. We're through to acetyl-CoA, or not the CoA, but at least an activated acetyl group in just a few steps, through to glycine pretty quickly as well. From acetyl-CoA into pyruvate, more CO2, more hydrogen, CO2 in green here. Pyruvate through to oxaloacetate, more CO2, and alpha-ketoglutarate. This is the Krebs cycle. Not the Krebs cycle as most people know it as an oxidative process, but the reverse Krebs cycle, or these first steps, are basically taking hydrogen and CO2 from the environment, reacting them together to make Krebs cycle intermediates. And these are used to make amino acids, aspartate, glutamine, glutamate, and so on. They're used to make sugars from pyruvate through phosphoenol pyruvate and up to ribose phosphate. And they're used to make an energy currency, in this case, acetyl phosphate. Now, the red boxes here, these are the precursors for nucleotide synthesis. So again, we're beginning to make purines and pyrimidines really very close to the center of metabolism. And the very core of metabolism from this autotrophic point of view is just hydrogen and CO2 producing these carbon skeletons right at the heart of metabolism. And most of it, almost all of these steps are actually exothermic. They will happen spontaneously. If you start with hydrogen and CO2 and warm them up under alkaline conditions, warm them up to 50 degrees centigrade, as you can see across the x-axis here, the total cell biomass releases energy. This is the free energy. It's quite strongly exothermic. If we shake hydrogen and CO2, we should get cells dropping out. Thermodynamically, that should happen. Of course, it doesn't happen. We're most favored of the amino acids, the fatty acids. You can see at the top, nucleotides cost some energy to make. But this idea that you shake CO2 and hydrogen, and really the energetically favored state is cells, is hard to fathom, but it's the truth. The reason it doesn't happen spontaneously is this barrier here to the reaction. To get through the first couple of steps up to formaldehyde, it's uphill. And then after that, it's downhill all the way. And this is the kind of the redox zone of organic chemistry inside cells. It's just slightly lower than CO2 itself. The methanogens are lowering this barrier by using a proton gradient. And that's what is driving the reaction between CO2 and hydrogen. Now, the question is this something that could happen in a prebiotic environment? And the answer is yes. It's just simple physical chemistry. The problem here is that hydrogen, it's just, if you don't like these reduction potentials, just think of this as uphill. So hydrogen can't reduce CO2 to formaldehyde. It's uphill reactions. It's not favored. The reduction potentials are not favoring it. But that's at pH 7 or any other pH. In the vents, hydrogen is bubbling out of the ground at pH 11 in these alkaline fluids, and that makes it much more reactive. The reduction potential is much lower. And CO2 is coming in, percolating in with the acidic ocean waters, these early acidic oceans. And in an acidic environment, CO2 is more reactive. You can think of it really easily. If a hydrogen pushes its electrons onto something else, what's left behind? The protons are left behind. In an alkaline environment, the protons react with hydroxide ions to make water. This is just a fizzing neutralization reaction that most people will have done in school, possibly in their mouths even. There's no way you can stop neutralization happening. The more alkaline the environment, the more favored that step is. In an acidic environment, you're releasing protons into the acidic environment. CO2 picks up an electron, it's got a charge. If there's a proton available, it balances the charge. It can now pick up another electron. It picks up another proton. It's easier to reduce CO2 in an acidic environment. And this is just formalized in the Nernst equation. The difference is about 60 millivolts per pH unit, and that's all I'm drawing here. So what we have in these vents, hydrogen in alkaline fluids is more reactive. CO2 in acidic fluids, acidic ocean waters, is more reactive, so long as they stay as two phases separated by some kind of a trifling barrier, a membrane of some sort, then there should be reaction. This membrane needs to separate and link. Remember what Peter Mitchell said. It needs to separate and link the two different phases. There needs to be transfer across this. It needs to frustrate the mixing, but be transfer of electrons or protons across the barrier. So I can imagine I'm now going to suggest a mechanism, and then I'll talk about how we can test it in the lab. I can imagine that here is a pore in a vent, and I've got protons, for example, crossing this barrier and driving the reaction between hydrogen and CO2. And the protons are perpetually being fed in from the oceans, hydroxide ions and hydrogen perpetually fed in the hydrothermal fluids. And if this is not formaldehyde, this is supposed to just be organic molecules in general. If that continues, the logical end point is we end up with something like a protocell with, say, fatty acid membranes surrounding it. So this is how I can imagine the process might work. Question is, am I mad? And the answer is, well, quite possibly. You can ask me that later on, but we can at least test it. We're now into science, and I've been trying to test some of these ideas for more than a decade. This was one of the first reactors that we built, and you can see it was very homemade and it didn't work very well. We tried precipitating all kinds of vent-like structures inside this, and they didn't work very well either. We ended up doing a microfluidics, and that's worked somewhat better. But ironically, it was a former student of mine, a PhD student, Victor Soho, who left my lab and finally nailed this a year or two ago. And this is in the left-hand side here. This is a schematic of the barrier. You can see that there's hydrogen in the alkaline fluids transferring its electrons onto the barrier, which cross the barrier to reduce CO2 at the other side in an acidic solution. I won't go through the NMR data with you, but basically what it's showing using 13C NMR and deuterons, 2H, is that it's the CO2 which is being reduced, and it's being reduced by protons that are coming from water on the acidic side. It only happens at a modest pressure, 1.5 atmospheres of pressure. It requires a pH gradient, so it really does require this pH difference. If you don't have that pH gap there, then it doesn't work. If you don't have the barrier, it doesn't work. If you don't have hydrogen gas, it doesn't work, and so on. So it requires this topology, it requires this setup. But these experiments weren't clear. Is it protons or is it electrons crossing this barrier? We couldn't really nail which way it's going. And I've been trying to do that. And one thing that struck me early on is that this is a microfluidic chip, and we've got acid flow on this channel. We've got alkaline flow in this channel. It's laminar flow, and so there's not much mixing going on here. There's no barrier here at all. This is just the mixing zone. And if we add ferrous chloride and sodium sulfide, then we precipitate an iron sulfide barrier between the two, and it can be quite a diffuse, wispy kind of barrier. And we notice that it tends to be acidic. So this is fluid inclusions trapped inside the barrier, and they tend to be acidic. So we looked at that a little bit more closely, and here you can see that this is the alkaline phase, this is the acidic phase, and this is a wispy barrier again between the two. And it's acidic right up to the very edge. We did some stop flow experiments, and we found that protons cross this barrier about 2 million times faster than hydroxide ions go the other way. So these barriers really do transfer protons. It's probably to do with the structure of the trapped water in the barriers themselves. But this at least is consistent still with this idea that we can have protons crossing a barrier and entering some kind of protocell. But I say protocells. This is in an extreme environment. This has got pH 5 or 6 in the oceans. It's got pH 10 or 11 down here. It's modern ocean salinity, almost certainly. The early oceans were pretty saline. And there were divalent cations, magnesium and calcium and so on. These are supposed to play havoc with these vesicles or protocells made from fatty acids. They should just collapse and fall to pieces. So we wanted to see, first of all, is it even possible to have vesicles under those conditions that are stable? Now, there has been some work done by Tom McCollum and others under hydrothermal conditions using a process called Fischer-Tropsch type synthesis. And what you're seeing here is long chain hydrocarbons with 15, 16, 17 carbons in the chain. And these are alkanols, so fatty alcohols, and fatty acids, alkanoic acids with a carboxylate group. Again, 10, 11, 12 carbons in a chain. And you get these quite complex mixtures. So we use those mixtures to try and make protocells from them. And you can see them here. This is with a fluorescent dye to show the vesicles themselves by confocal microscopy, and this is cryo electron microscopy, and you can see that we're getting vesicles. I'm not going to go through the details of this, we can come back to it, but to save time I'm going to skip over it. But we're getting stable vesicles at pH 12, 70 degrees centigrade, 600 millimolar sodium chloride, which is equivalent to modern oceans, 50 millimolar magnesium chloride, 10 millimolar calcium chloride. So really harsh conditions, the kind of conditions I was saying, they're supposed to fall to pieces. And if you just use a single fatty acid like decanoic acid, they do indeed fall to pieces. But when we have these more complex mixtures, they're quite robust. So this was really pleasing. We can see stable protocells in these environments. And even more pleasing, the kind of the proteins that are driving the reduction of CO2, I mentioned a couple of iron sulfur proteins, ferredoxin and the energy converting hydrogenase. These are iron sulfur proteins that contain 4Fe, 4S clusters. And we're wondering, in a prebiotic environment, I can imagine precursors, mineral-like precursors to these, but can we get to 4Fe, 4S clusters? So we simply tried mixing cysteine in water at pH 9 with ferric chloride and sodium sulfide, and we get the characteristic shoulder on UV-vis spectroscopy. And it's concentration dependent of the cysteine. It depends on the concentration of the iron and the sulfur as well. Mossbauer spectroscopy confirms that these are 4Fe, 4S clusters, and we have also 2Fe, 2S clusters and some other species in there. And cyclic voltammetry shows that they are redox active. They're not as reducing as I would like them to be. This is a silver electrode for those who like their electrochemistry. So minus 400 millivolts is not very reducing, certainly not reducing enough to reduce CO2, but this is probably a complex peak, and we need to try and work out what else is in there. We think there's several species in there, so we're still working on that. But the key thing is just mixing monomers, cysteine, iron, and sulfur in solution, and we're getting 4Fe, 4S clusters, the same as life. I'm just going to show you two slides on some slightly more complex chemistry that we've been working on. If these ideas are right, then the whole of metabolic biochemistry ought to be happening spontaneously under these conditions. People haven't done much work on this. I mentioned at the beginning, lots of people have worked on cyanide chemistry and so on. This is following the pathway of pyrimidine, of uracil synthesis that cells use, and this is doing it in the absence of enzymes, just using metal ions as catalysts. We have a bunch of metal ion catalysts, and curiously copper works quite well. We're able to generate uracil. We can change the conditions, switching pH, changing pH. We were able to optimize the conditions, and the conditions that worked best turned out to be basically alkaline hydrothermal conditions, pH 9, 90 degrees centigrade, 100 bars pressure, modern ocean salinity. It worked around about 10-fold better, in fact, than the conditions in which we started out. And this one I thought you might like. This is ATP. If we start with ADP in solution and we add acetyl phosphate, we're able to phosphorylate the ADP to ATP. Only in the presence of ferric iron, funnily enough. Other metal ions do not catalyze this reaction. This is just in water. There's no enzymes, no other catalyst there. And we get 15 to 20% yield. Carbamoyl phosphate can do it a little bit. This is at pH 5.5 to 6. Works less well in alkaline conditions. And we tried a whole range of different phosphorylating agents, and really only acetyl phosphate and carbamoyl phosphate worked at all. But this is the really beautiful thing that I wanted to share with you. Phosphorylating ADP to ATP, we get quite a nice peak. That's what we're seeing here. But if we try to look for ITP or GTP or CTP or UTP, we're getting no more than the control values. In other words, there's something favored in water about ATP itself. This is why, in my mind, why ATP is the universal energy currency. You can phosphorylate ADP to ATP in water with ferric iron as a catalyst using acetyl phosphate, which is still used in bacteria today as the kind of the linchpin between thioester metabolism and phosphate metabolism. So it's really, it's beautiful that it happens spontaneously in very simple prebiotic conditions. So just to finish for the last 3 or 4 minutes, if I may, I want to just talk about where it goes and how the code might emerge, the genetic code in these conditions. Have I got 3 or 4 minutes, Win? I hope I do. So This is modeling, this is mathematical modeling, and we're considering a protocell sitting in a barrier here, fixing CO2 using iron, sulfur, minerals trapped by organics, so amino acids like cysteine, reducing CO2 to an intermediate like a thioester, methyl thioacetate or something like that, equivalent to acetyl-CoA. We're branching from there into amino acids, sugars, an energy currency like acetyl phosphate, and fatty acids. So the fatty acids would partition straight to the membrane. The amino acids can interact with the iron sulfur clusters and drive CO2 fixation and so on. What happens if we allow this chemistry to move forward right the way through to nucleotides, as I've just shown you seems to be possible? And then we assume that there's a positive feedback. We can assume a positive feedback on CO2 fixation. This is what we see with folates, for example, and molybdopterins in methanogens. They're involved in CO2 fixation. Whoops. So we can assume a positive feedback there and ask what happens. Or we can assume all kinds of other positive feedbacks on any of these pathways, see what happens then. Or we can assume autocatalysis just favoring nucleotide synthesis. And what we find, and I'm not going to go through all the results here, but what we're finding really is that yes, it can drive protocell growth under some sets of conditions. If we have this situation where it's just, for example, favoring amino acid synthesis, that tends to collapse growth. We don't want good catalysis of particular pathways because that forces flux down one pathway at the expense of the others and collapses protocell growth. So we actually want bad catalysis, general catalysis, the kind of things that nucleotide cofactors still do today. The other thing we kind of naively hoped was that we'd be able to accumulate, some set of conditions would allow us to accumulate nucleotides at high concentrations inside these protocells, and then they would be able to more likely to spontaneously polymerize. But the assumption that they're catalysts means that actually they never do that because they catalyze the growth of cells, and so they keep dividing and diluting the contents at every cell division. So in principle, this can happen. How could genes emerge in this environment? So this is just what I'd like to leave you with, and I'm going to skip lightly over it and you can come back and ask questions later on. There are patterns in the genetic code, as I'm sure many of you know, and I'm just going to point to 3 of them very quickly. This is a quick scheme of autotrophic metabolism, what I showed you earlier on. And in gray, this is where the first base of the codon is a G. And in yellow, this is where the first base of the codon is an A. So the first steps are purines, encoded by purines. And then with green, this is uracil is the first base, and with blue, it's a C for that first base. So we see that there is, this is the minimum number of steps from CO2 fixation. And there's a very clear relationship of the order of the bases. It seems to suggest that purines came first. Purines came before pyrimidines, and especially G was early. This is the second base in the codon. And if we have a G or an A at the first position, we see quite a strong relationship. This is from 43 different indices of hydrophobicity. And what we're seeing here is quite a strong correlation between the hydrophobicity of the amino acid and the base at that second position. It's not nearly so strong if there's a pyrimidine at those first 2 positions. So there seems to be both a time factor and a biophysical interaction going on here. And this is the final thing I'd like to leave you with. There's also patterns at the 3rd position in the code, which we think other people haven't spotted before, rather surprisingly. So this is an inverted codon table where at position 3, we either have a purine or a pyrimidine. At position 2, up here, we have the pyrimidines. Down here, we have the purines. And this is position 1. This is the first position of the codon. So if we have a G and an A at the first and the second positions, and a G and an A at the first and second positions, it matters whether there's a purine or a pyrimidine at the 3rd position. It's either we can predict that it's going, if there's a purine there, it will be glutamate. If there's a pyrimidine there, it will be aspartate. And that difference depends on the size of those amino acids. So again, there's a physical relationship between the size and the hydrophobicity of amino acids, and probably a time factor. We can't understand the details of this yet. But all I need to do is posit that there are non-random relationships between the amino acids and random RNA sequences. So if we have biophysical interactions between amino acids And random RNA sequences, if we're introducing random RNA polymers into these growing protocells, there's no information here yet. It's a purely random sequence, but we have a non-random peptide through templating. And in the context of growing protocells, that has function. If it's a hydrophobic peptide, it's likely to go to the membrane. It may be involved in CO2 fixation. If it's a hydrophilic peptide, it's likely to chelate ions like magnesium. Aspartate would interact directly with magnesium, and the RNA polymerase is a magnesium-dependent protein, which is always aspartate chelating magnesium. It's just biophysics. All kinds of mixed peptides are likely to bind nucleotide cofactors and promote protein metabolism. We can easily see how functions can emerge in a growing protocell system just simply through biophysical interactions. And that means that we have sequence-dependent functions, and they will be inherited so long as that sequence is copied. And so information comes into biology right from the very beginning in this protocell context. And I don't think it does in any other context. So I'm going to leave you with that. And I have to say thank you enormously to my group, because they're brave people to risk their careers coming to work with me on questions like the origin of life, where there's no guarantee they're going to find anything at all. So I thank them, and I thank you, and I'm going to pass over to Jennifer. And I think the questions come later. Thank you. I hope I didn't overrun too much.
So please remember to continue to submit your questions, and we will have a question and answer at the end of Jennifer's talk. So now we're going to turn to the emergence of the eukaryocyte. Jennifer?
Yes, let me get into. So can you see my PowerPoint or not?
Not yet.
Okay, so I've got to, let's start. I've got to share my screen. Can you see the presentation?
Yes.
Perfect. Well, it's quite an honor to follow Nick Lane, whose work I have really been in awe of. A lot of the ideas that I'm going to talk about today actually come out of his book, The Vital Question, which really transformed my thinking about eukaryotic cells and the way that they've originated. So this is going to be a perspective. Basically all the ideas that I'm going to be talking about are ones that we really need further testing. But I'm going to take the hint from Wynn to just throw out some of these ideas just to start a conversation. So I think one of the things that really forces you to really think hard about the origin of eukaryotic cell is to, first look out at a planet that has no life, like Mars, and then move into a planet like Earth that is full of life. And when you look out at this planetary landscape, what you're seeing is hidden underneath plenty of prokaryotic life, but also an incredibly emergent eukaryotic system. That's what we're looking at here, and that's what we all are operating within as physical big human beings. Now, the evolution of there's a lot of thinking about not only the tree of life, how we move into this large eukaryotic world, but oops, sorry. I think one of the things that is really sort of a starting question is, first, why did it take 2 billion years before we reached this site of the last eukaryotic common ancestor? And then why, what are the changes that happened over the next 2 billion years to create this incredible diversity and structure that we see that's characteristic of eukaryotic cells? And I think one of the clear points that Nick's presentation really made clear is that in the prokaryotic kingdom, it's really metabolism that is at the heart. Basically, life was getting hold of a rich metabolism that ultimately led to a new invention. This invention is the eukaryotic cell, which then moved into a different type of dimension in terms of exploring space on a planet, and that is structural diversity, where we have animals, plants, fungi, seaweeds, and amoeba. Now, one of the things that has always excited me about eukaryotes as opposed to prokaryotes is the complexity of their internal compartmentalization. What you're looking at is an EM slice of a mouse fibroblast, and you can see the huge diversity of subcellular structures or compartments that comprise this cell system. Now, what's quite amazing about eukaryotic cells is that even simple, more sort of compact versions of eukaryotic cells, including this parasite, Toxoplasma gondii, has the exact same set of 9 organelles that characterize this much larger fibroblast. So basically, there's a conservation in the compartmentalization of these eukaryotic cells, which I think deserves thinking about when we start coming up with models for how the eukaryotic cell itself evolved. Basically, all eukaryotic cells have conserved membrane-bound structures that are performing very specific tasks, which range from energy metabolism in the case of mitochondria to delivery of macromolecules to the outside of the cell to different other subcellular compartments, and also a process of endocytosis and uptake of macromolecules. Now, there are several models for the origin of eukaryotic cells, but 2 of those I'm going to focus on today. And perhaps the most widely recognized model for the origin of eukaryotes is what we call stepwise evolution. And basically, the idea behind this model is that prokaryotes basically got tired of being just a simple prokaryotic and started elaborating its internal membranes, potentially by invagination of the plasma membrane. But basically, that led to what we call a proto-eukaryote, which already had a simplified set of endomembranes, already had sort of perhaps a primitive nucleus. And basically, all that was needed is to create a more modern eukaryotic cell was the internalization or uptake of bacteria, which would create a mitochondria, which itself could become an integrated part of that eukaryotic cell by transferring part of its DNA into the nucleus of this proto-eukaryotic host. And ultimately, you would then get the complex eukaryotic cell. Now, the problem with this stepwise evolutionary model is that there are no surviving proto-eukaryotes today that show any evidence for this pathway. We can't find proto-eukaryotes. We can't find eukaryotes that have a subset of the compartments that I just showed you, or proto-eukaryotes that lack mitochondria, or some hereditary vestige of the mitochondria. Basically, all eukaryotic cells now have very similar, identical sets of organelles. We also know that these organelles, are these internal organelles of these eukaryotes are all doing very similar things in all eukaryotes. So basically, these are arguments that doesn't rule out this stepwise evolution, but makes you start wondering whether some other model might fit the bill more specifically. And that other model that I want to talk about today, and really builds on the really fascinating ideas that Nick talked about today, is a type of metabolic symbiosis. And then this model basically is that a eukaryotic cell is fundamentally a symbiotic relationship between the two types of very primitive prokaryotes that Nick talked about, the methanogens and the acetogens. So the acetogens are producing carbon dioxide and hydrogen. They are the bacteria. And the methanogens are producing methane. And it's a collaboration between these two different prokaryotes that gives rise to a symbiotic system that ultimately moves into the creation of a complex eukaryotic cell. So let's take a closer look at this. And I don't know whether you can see, is there a panel up here that blocks people from seeing the title of these slides? I'm just curious. I can't get rid of it. So anyway, bear with me. Basically, this is just a zoom up of the this metabolic symbiosis model where we have these two different types of prokaryotes that basically, once they begin sort of interacting metabolically, and ultimately when the alpha proteobacterium, which is the acetogen, gets taken up into that methanogenic archaeon, what emerges is an increased energy dependence as well as potentially energy output in that system. And it's this energy dependence and output that I want to focus on right now. Basically, what's the basis of this increased energy dependence? And for that, I'm going to sort of move to what Nick talked about, which is this unexpected aspect of the energy of life, which is that cells are deriving energy from really one major type of chemical reaction, and that is flow of protons across a membrane. This, as Nick said, is by many people think is one of the most counterintuitive ideas since Darwin, but basically it was proposed by Peter Mitchell, and it's the cross-membrane protein gradient that Peter proposed drove energy metabolism within cells. In particular, he was using it to describe the mitochondrial respiratory chain and the way that it is producing ATP. Basically, these are the various enzymes, subunits that are part of this electron transport chain. But basically what they're doing is pumping protons across this membrane bilayer, and as a consequence, creating an electropotential gradient. Now, we know that all living, well, based on what Nick said, all living cells are powering themselves through this type of mechanism. So both archaea as well as bacteria use this type of chemoosmotic coupling to drive their energy production. So a question is whether there are differences in the way that archaea and bacteria are doing this that might provide clues as to why having a symbiotic relationship between archaea and bacteria might produce a system that can give emergent properties to life, i.e. a eukaryotic cell. So let's look at the two different ways that archaea and bacteria are doing this. And one of the things that I think sort of pops up is if we look at the phospholipids that are characterizing archaea versus bacteria, Nick mentioned that they are different. Yes, indeed they are. But what's really interesting is the difference between the linkage of the phospholipid side chain, the acyl side chain to the head group. In the case of archaea, it is mediated by an ether linkage, whereas in the case of bacteria, it's an ester linkage. And that turns out to potentially be extremely important in terms of regulating the flow of protons across bacterial membranes. Recently, Maria Mencia, a theoretical and a biochemist, has proposed that with ester linkages in lipids, the protons could potentially flow more fluidly through the membrane, the inner part of the bilayer, in contrast to phospholipids that have ether linkages, where you don't have the ability to just flow these protons across the membrane. And that would facilitate the ability of the enzymes in the electron transport chain to be able to be coupled, to flow protons fast, to drive fast proton flux. This could be, and this is very speculative, but this difference in proton pumping efficiency, it might help explain some of the differences in archaea and bacterial habitats and characteristics. We know that, at least today, archaea, if you want to find them, you got to go to extreme environments, either these sulfur ponds or these hot pools. And if you try to grow these archaea, you find that they typically are extremely slow growing. Bacteria, on the other hand, grow extremely fast, and you can find them all over the place. This is just in the soil. Basically, you pick up a pot, some soil, it's just full of bacteria. So that raises the question of, okay, well, if bacteria is so good at growing and can flow protons really fast to give you a lot of energy, why didn't a bacteria become a eukaryotic cell? Why can't we just scale up a bacteria to make it like a eukaryotic-like cell? And part of the problem with that is that the bacteria has a very small amount of DNA, and that DNA is largely devoted, or a large chunk of it is devoted to driving the production of the proteins that are comprising the electron transport chain on its surface for energy production. So the thinking is that the bacteria's genome is just too small to support the energy needs of a cell that swells up. Basically, you're not going to get this DNA to be able to couple itself in terms of its production of proteins, its transcription and translation to match what's going on in this large system. Well, why not just put a whole bunch of bacteria DNA into, why doesn't the bacteria just swell, but also just make a whole bunch of copies of its genome? And this also has a problem because this small genome also, we know it could be the proteins that ultimately are going to be read out from this genome, again, have the problem in the case of proteins that are involved in the electron transport chain of finding this membrane to drive proton flux, to drive energy production. So basically what the answer seems to have been in the case of eukaryotic evolution is to keep the small size of a bacterium that is really supporting the energy needs of the cell, but it just inhabit a larger environment. And this is basically what the eukaryotic cell is. It's a bigger cell that's run by multiple power units, which are mitochondria. So each of these mitochondria have their membrane close around them. They're churning out proteins that are going to drive the energy production for this eukaryotic cell. So this type of thinking has some implications. One implication is that the proteobacteria that it has been taken up by the archaea, which is the mitochondria, must or is likely to have been a major driver for evolution in the eukaryotic system. Basically, that mitochondria is going to really be regulating a lot of things. And is that the case? Well, and we should see a lot of features of mitochondria all over, or marked all over the way the eukaryotic system is working. And one thing that we immediately know is that eukaryotic membranes, and I wish we could get rid of this panel here because you can't see the title here, but basically this is just a small micron volume of a eukaryotic cell where we've segmented out all of the different organelles within this system. And what I want to just draw your attention to is that the membranes that comprise all of the eukaryotic cell are all ester linked, like bacteria. They're not ether linked. So the ester linked feature of phospholipids found in bacteria has been embraced by the eukaryotic cell. All its membranes are ester linked. There's none that are ether linked. So basically the bacteria sort of pushed out any archaea type lipid that was ether linked during this evolutionary process. We also see the preeminence of mitochondria in cell physiology. This is just live cell imaging of mitochondria in a U2OS cell. You can see that mitochondria are just distributed all over the cytoplasm. They're highly dynamic. If we then interrogate all of the features of these mitochondria in terms of their functionalities, we can see that basically almost all aspects of cell physiology really have mitochondria at their core. So for instance, all the building blocks of the cell, whether it be nucleotides, proteins, lipids, or heme, are all made from the fundamental TCA cycle operating inside that mitochondria. The mitochondria also can give rise to lipids. It can store lipids. I mean, it's taking in fatty acids and glucose through this glycolytic pathway, which feeds into this TCA cycle, but you can move out to store lipids. And I'm going to be talking about this later in my presentation, which plays a critical role in the way that cells can handle starvation and deal with very dramatic changes in availability of nutrients. The mitochondria also is controlling cell death. It's controlling signaling. It has it's a calcium storage depot. Basically, all the characteristics, all of the macromolecules that are, and a lot of the metabolism of the cell really are centered around the mitochondria and the way it's functioning. So basically, all of this is suggesting that the eukaryotic cell arose through a single endosymbiotic event between an archaea host and this mitochondrial, it's a bacterium, alpha proteobacterium, that ultimately is going to give rise to the mitochondria. So that then raises the question of how does eukaryotic cells then get big genomes, and how do they become morphologically complex? Is there a way that we can start thinking about that to get a better understanding of, we have this new type of synergy between a sort of a metabolic symbiosis, if you will, between these two different types of prokaryotes that's given rise to this new form, this eukaryotic cell. How does it get a big genome, and how does it become morphologically complex? Well, the bigger genome can easily be sort of imagined by basically being powered by all of these mitochondrial ATP power units. A lot of the mitochondrial genome, a lot of the alpha proteobacterial genome that was originally found in these endosymbionts, we now know have moved into the nucleus, making this a large genome. This genome became even larger because it basically is the home to all of the archaea-based genes. So basically, it's not hard to imagine how you could create a bigger genome run by multiple power units to create this, to enable the eukaryotic cell to operate in that regime. But what's not so clear is the emergence of the morphological complexity of all of these membranes and compartments inside the eukaryotic cell. And that's what I want to talk about for the next 5 minutes or 10 minutes of this talk. Basically, what I'm going to propose is that the patterning of lipids are playing a very critical role in this type of compartmentalization. And basically, the emergence of this internal complexity inside these eukaryotic cells was enabled by the presence or the emergence of particular types of eukaryotic lipids. So eukaryotes have a combination of glycerophospholipids, sphingolipids, and cholesterol, which neither bacteria or archaea have. In particular, archaea do not have sterols or cholesterol in the way that we modern eukaryotes have. And it's the presence of that cholesterol which is giving rise to a very interesting characteristic of membranes, which is their ability to partition into regions where the sterol can order the sphingolipids to create a thicker bilayer that into which different proteins preferentially sort. Now, there's a lot of interest in this in the evolution of sterols in eukaryotes. Basically, bacteria brought in some precursors for sterol evolution, but really it took off very early in the evolution of eukaryotes, this sterol. And modern day eukaryotes that can't make sterols will bring in sterols from other sources. So sterols are absolutely a fundamental part of eukaryotic membranes. So I mentioned how these sterols or cholesterol can order these membranes. Basically, phospholipids are highly disordered, they're floppy, makes any membrane, the membranes that contain just pure phospholipids are leaky and highly disordered. As soon as you start adding sphingolipids and cholesterol, especially the cholesterol, you can begin ordering these membranes. And you can see this experimentally by just taking these three classes of lipids, putting them in a vial, shaking the vial up, and you'll see if you create vesicles, we call them giant unilamellar vesicles, and drop the temperature, you can see that these vesicles will phase separate into ordered and disordered domains, where the disordered domains will be primarily phospholipids, and the ordered domains will be primarily sphingolipids and cholesterol. It turns out that we can see this even in biological membranes in cells. You can hypotonically swell out, bleb out a vesicle from your cell. And if you lower the temperature, you can see spontaneous phase separation using lipid probes of that bleb into a disordered and ordered domains. We've recently introduced fluorescent proteins that have been proposed to target to ordered versus disordered domains onto the plasma membrane of cells. If we bleb out some of that plasma membrane and lower our temperature to drive phase separation, you can see that different types of proteins, this is transferrin receptor, this is EGF receptor, they phase partition into different domains in these plasma membrane vesicles. This is extremely interesting and exciting because what this is saying is that the presence of these lipids allows for very complex sorting of proteins on biological membranes in eukaryotic cells. It's not clear that this is possible to this extent in the prokaryotic kingdom. Now, I want to just share with you some real biology that we think underlies this, that involves this phase partitioning. This is a yeast vacuole from a yeast that is well fed. And you can see that when we introduce a marker, in this case, GFP-Pho8, which likes being in a liquid disordered membrane, as soon as we starve this yeast cell, we can see that the vacuole will phase partition into this buckyball-like pattern or soccer ball-like pattern, where the disordered membrane is now separated from these black areas here, where the membrane is enriched in sterols and sphingolipids. And this is just showing this is a freeze fracture of that vacuole from that starved yeast cell. This is the dynamism of this lipid sorting that's happening in that yeast, that vacuole. And this is a real cell where we're looking at this. Now, one of the things that we've done in terms of trying to analyze how this phase partitioning occurs and what its purpose is shown here. Basically, when the cell is starved of glucose, autophagy is initiated, and that brings membranes from the endocytic pathway that are in sterols to the vacuole. Sterile abundance in that vacuole, triggered by this fusion of the autophagosome, starts driving phase partitioning on that vacuole. And that then leads to the ability of the vacuole to start making contact with particular organelles, in particular the endoplasmic reticulum, which itself can make contact with the mitochondria. And basically, what we're proposing is that this enables a lipid pipeline to flow into the mitochondria under starvation conditions. Because what we find in these yeast cells is that when they undergo starvation, they are very happy. They can survive, 50 days under acute glucose restriction. And what happens is the mitochondria actually proliferate dramatically, as you can see under these conditions. Cell respiration is boosted. As you can see in this map, we look at oxygen consumption under this acute glucose starvation regime, in contrast to cells where we have not initiated this type of response in terms of phase partitioning and the ability of these organelles to begin to communicate with each other. So basically, this is just an example where we see that the cell, in adapting its pathways for dealing with different types of nutrient stresses, the mitochondria is playing a key role. There are pathways that are engaged to specifically feed fatty acids into that mitochondria and also enable that mitochondria to grow its membranes in order to give the cell ATP under these nutrient restricted regimes. So basically what I'd like to propose is that the appearance of cholesterol and sphingolipids in membranes could be what might be called an event horizon for eukaryotic cell evolution. And it's enabling because it's allowing membrane compartmentalization and trafficking. Basically, it's only by being able to partition these membranes that you can get preferential sorting of proteins. Now, the support for this is that we know that bacterial membranes, it's only bacterial membranes that have sterile-like lipids and sphingolipid-like molecules. Archaea don't have any hint of that. Neither bacteria or archaea have true sterols in their membranes. Basically, we know that mitochondria are collaborating with other organelles in cholesterol biosynthesis, and that the cholesterol and the sphingolipids are creating these lipid gradients. So some membrane-centric speculation. My thinking is that basically with the ability to phase partition membranes, the primitive GTPases and other proteins that prokaryotes had, including escorts, would then be able to rapidly evolve. So basically, a lot of the the GTPases that are shaping and driving, polymerization of various cytoskeletal components, they basically are very much coupled to being able to associate with membranes. And this phase partitioning may be a very important part of that process. And so we think, it's also possible that this could have led to the evolution of cytoskeletal nucleators, many of which we know, like ARP23, bind membranes. Indeed, we know that membranes like the Golgi apparatus can bind gamma tubulin and create microtubule asters. So a lot of the orchestration of cytoskeletal organization within the cytoplasm of a eukaryotic cell is very much dependent on specialization of membranes that will either sequester or accumulate different types of lipids that recruit nucleators for cytoskeleton. So we think this may help explain why prokaryotes never evolved more complex cytoskeletons. They had neither the specialized membranes nor the array of nucleators to do this. So my last question is, well, how can we address these issues? How can any of what I've talked about today be proved or in any way supported? Basically, I think we're going to have to address this in a lot of different ways. One is we need to do tools to visualize, probe, and measure proteins and lipids within the cell. We need to really look at this phase partitioning and see exactly what it's doing. We need higher resolution to examine protein distributions in relation to lipids within organelles. And finally, mathematical modeling would certainly be incredibly valuable to assess the dynamics of the system and really start leading to other types of hypotheses for testing some of these questions. So with that, I want to thank you, and I will now stop sharing, and we can be open for questions.
And thank you very much, Jennifer. And Nick, we do have a series of questions, and I hope that both of you are thinking of questions you would like to address to each other. One question that's been asked, Nick, is whether could life perhaps have been started a few times, more than one abiogenesis? Is there an idea that bacteria, archaea, and the eukaryotes may have arisen several times, or have arisen from?
I mean, they may have done. There's no evidence to support that. I mean, they plainly life shares a common ancestor, because the genetic code really is the single strongest piece of evidence that suggests that all life is derived from the same source. Now, right at the end of my talk, I suggested that there are biophysical reasons for the code. I don't think that would explain the whole code, and I would be absolutely amazed if we found bacteria on Mars or somewhere like that, and they had exactly the same code, and we concluded that they didn't come from Earth, or vice versa. It would be shocking if you like, chemical determinism can take us right up to the code itself all the way. It's possible it can, that we don't know enough yet, but I would be surprised. Now, that doesn't mean to say that there weren't multiple origins of life and that it simply disappeared and there's no evidence for it. And what would you see in a fossil record? You'd see smears that look a bit like bacteria in the rocks, and you'd never know what they were. They would have organic matter in them. There was no phylogenetic evidence for multiple origins, but all that really means is it didn't survive. So it's very hard to say. I think in the case of bacteria and archaea, there's almost a more difficult question about if they shared a common ancestor and they, let's say, emerged separately from vents, then why did it only happen twice? Why did it happen? Why did they diverge once and not multiple occasions, or not diverge at all? And I think I have some thoughts on that, but they're not terribly well thought through. But it's really to do with the origin of pumping. As soon as you can pump, then you have a tremendous advantage over cells that don't know how to pump. And so I think that as soon as you get that's the last step, if you like, of becoming a free, independent individual cell, is you can now leave a vent and you can live somewhere else. So as soon as cells had left, then I think Darwin was right. I mean, basically, he said once life has appeared, then it's going to eat everything else. It's going to be really difficult for a kind of a prebiotic system in a hydrothermal vent to get a foot in when there's already cells out there. They're so much better at what they do. Jennifer, do you have thoughts on that?
Didn't Darwin say that he thought that life evolved in a nice, calm little pond, a warm pond?
Yes, he did. I think he was mistaken on that one personally, but there are most of the chemists who favor the cyanide chemistry are very fond of quoting Darwin and saying the great God Darwin almost said life started in a terrestrial pond, therefore it must be true. And they quietly forget about some of the genetics that he didn't know about and so on. I mean, Darwin was a genius, but he wasn't right about everything.
Right. So I have a question. Oh, I was just going to ask Nick a question, although this may take us into a different world. But one of the things that was exciting about your reactor that you generated is that it's able to remove CO2. Basically, it's pumping CO2 out of a system. Have you thought about using that at all for sort of scaling up for global warming purposes?
Absolutely. I mean, first of all, we have to make it work.
Yeah.
Right now, it works a little bit, but not very well. But it's basically, I think one of the reasons why these ideas on CO2 and hydrogen are coming more into vogue than they were 10 years ago, 20 years ago, when all the chemistry was cyanide chemistry. And now there's more interest in, can we make CO2 react with hydrogen? And it's really about global warming and energy security. Because if we can strip CO2 from the atmosphere, split water, take the hydrogen, make hydrocarbons, and effectively have a carbon neutral economy where we don't have to change so much, that's an ideal solution to everything. And that's what these cells in these vents do. That's how they grow. And you know, there has to be a simple way of doing it.
Atmosphere too, yeah.
Yeah.
It's amazing.
Nick, you touched on something that we have several questions about. Would you elaborate as to how an early membrane developed pumping activity to maintain internal and external milieus?
Yes, I don't need that to happen early because in a hydrothermal system, we've got continuous flow. And so I suppose the key thing about these ideas is that pumping is a late step. But what that means is that without pumps, protons have to come in. If you've got a modern membrane, which is effectively close to being proton tight, a coupled membrane that we would see in the mitochondria or that bacteria have or that archaea have, that's the last thing you want. If you have a pore, a turbine, an ATP synthase or an ECH or any piece of molecular machinery, proton comes in and there's no pump to get it out again, then you've just collapsed the driving force. So what you actually need is a leaky membrane, leaky to protons specifically, so that they can come in and then they're swept away by the flow of alkaline fluids. They react with the hydroxide ions. So it can only work if they don't have a modern membrane, a modern phospholipid membrane that looks like an archaeal membrane or a bacterial membrane. They have to be more or less leaky to the outside world for these ideas to work at all. And so that would be my explanation for why bacteria and archaea have quite different membranes. They arose relatively late because they arose at the same time as pumping. Now, so how did pumping begin? I think we are dealing then in the world of genes. We're dealing with proteins which are encoded by genes, which are capable of conformational change. We're dealing with molecular machines. So I personally doubt that there were sophisticated pumps before genes. There are ways of doing it. Quinones, for example, can pick up protons and carry them across the membrane. And it would depend where they go. They can shuttle backwards and forwards depending on the redox difference and depending on the pH difference. So it's possible to have pumping in the absence of complex proteins. But if you've got a system where effectively you don't need it because the vent is doing all the work, the vent is keeping the gradients there, then I kind of don't need pumps until the end, where cells effectively in the end need to leave or are cut off from, there will be a selective advantage if you're in a corner of a vent where there's not that much flow and you've figured out a little bit how to pump your own protons out, you will thrive in that region, whereas other cells wouldn't. So it's easy to imagine selective advantages. It's really difficult to prove any of this stuff. We're kind of stuck with the central questions that we can actually test.
So could you have an exchanger without having a protein? Is that what you're saying?
Yes, you can have an exchanger without having a protein so long as it's a semi-open system. Stuff has to come in and leave again. In other words, I mean, from an origin of life point of view, it's actually very good. the last thing you really want to have is to say, right, I need a really sophisticated membrane and a molecular machine at the origin of life, otherwise nothing is going to work. What I'm actually saying is we need a crappy membrane, and we don't need any molecular machines. We just need continuous flow in a hydrothermal vent, and that does all the work for you, and that's just powered in the end by gravity. So we need bad systems, and we need to see ways of how they will get better, why they will get better, and those steps. So all of this up to the level of, as soon as we've got genes, where I finish my talk with the beginnings of random RNAs, but non-random templating of peptides, then we have natural selection as we know it. And so long as you have the power to keep generating these things, so long as you can begin to copy them, then everything changes. That's when life becomes living, to my mind. Before that, it's prebiotic chemistry.
So it becomes an RNA world from the beginning at that point.
But it's an RNA world inside growing protocells that are capable of doing all the metabolic biochemistry that other cells can do. And that just solves, to my mind, all the problems. But, you know, lots of people would argue with that. But it makes a lot of sense. The question is any of it true? The beautiful ideas killed by ugly facts all the time.
We have one question about what's the chemical basis for the generation of the alkaline hydrothermal vent? Why are they alkaline and not neutral or acidic?
Well, it's basically a reaction between the rock and the water, between olivine, which is rich in ferrous iron, and essentially the ferrous iron is reacting with water and becoming a ferric hydroxide, and some of those hydroxide ions are simply being released into solution. And that's where the hydrogen is coming from. The electrons are coming from the iron as it's being oxidized, and the water is being split into hydroxide ions and hydrogen in effect. So that's why it's alkaline. And it happens on any wet rocky planet. They're always alkaline. It's the only system that we know of really that generates alkaline fluids.
So do you think there are similar vents on other bodies in the heavens?
I think all of them, probably. I would say any wet rocky planet will have these vent systems on. And that's one of the pleasures of these ideas, is that it makes life potentially completely ubiquitous. Except then, as Jennifer was saying, we have this kind of 2 billion year gap before we see complex life. And so if we find life on Enceladus, or we find life, you know, somewhere else in the universe, the chances are it's going to be bacteria, and most people won't be very excited about it. You need the kind of processes that Jennifer was talking about to go from bacteria or archaea to complexity. Can I ask you a question, Jennifer? Because I'm curious to know what's the energy costs of having these phase transitions in membranes and being able to, all this dynamism that you're talking about, is it tremendously costly? Is it something that would be beyond bacteria?
The phase transitions require no energy. I mean, you don't have an energy input from an exogenous source. You need to make the membranes themselves, though. And the membranes themselves will spontaneously prefer being come together if there's cholesterol and sphingolipids, for instance. And the idea is the introduction of the proteins into these bilayers proteins will prefer being in the thicker bilayer from the sphingolipids and the cholesterol, or the stiffer, versus more prefer being in a more fluid lipid environment. And that's how you begin to sort proteins and in a bilayer. And once you start sorting proteins in a bilayer, you can start doing very interesting sort of further recruitments of things in the cytoplasm to build cytoskeleton, to create a vesicle that can move from one place to another. So it's really I see it as once you realize that the complexity of these lipids is not present in these archaea and bacteria, you start to understand why they can't do intracellular.
If they were present in bacteria and archaea, do you think that would allow bacteria and archaea to become larger, or are they still effectively stymied by the issue with mitochondria and the energetics of it. And so because eukaryotes can become larger, they now need more membranes in effect, and then can do more sophisticated things with them. So I'm just wondering if you're seeing it as the chemistry of the sphingolipids was beyond bacteria, or if it's just that they had no need for it because they could never expand to do that kind of thing.
I think
anyway.
they had no need for it, because I don't think they would be able to expand, because I like the argument that you make in terms of the genes that are coding for these electron transport components need to be near. They need to be regular. I mean, they got to be coupled to the expression of the proteins. And that just doesn't, I mean, because bacteria and archaea both have those electron transport system on their outer membranes, they're beholden to a small size. But the eukaryote doesn't have that on its plasma membrane. It's got energy packets, the mitochondria, for doing all that, which is just an amazing, cool way to, deal with the problem.
So in terms of where all these membrane systems came from originally, there's some lovely ideas about the outer membrane vesicles from mitochondria, but also budding from bacteria and from archaea as well. It's quite a common process.
Yeah.
But if you're doing that inside a proto-eukaryotic cell, then you're effectively producing at least bad membrane systems. You're producing vesicles inside cells that are going to fuse with each other, going to do things. I mean, is part of the problem, how do you stop it becoming a horrible mess?
Yeah. Yeah, that's a good question. I mean, the first thing you've got to shift over, I mean, so the archaea lipid just disappear. I mean, basically you got rid of it, so that now all the phospholipids are ester linked. So it's one system, and that's a huge argument against the classic model, the stepwise model for the origin of eukaryotes, because, basically that stepwise model proposes it's an archaea. It's going to have the archaea lipids that are making all of those internal membranes. And
yes,
none of the modern day eukaryotes have the ether-linked lipids.
I think there are some ether-linked lipids in eukaryotes, aren't there? I think occasionally, but they tend to be as
peroxisomes. Yeah, they're very rare, very rare.
Yeah, right.
Yeah, they're part of a pathway in peroxisomes for making certain types of big lipids, myelin, for instance. Yeah.
Right.
But yeah, I wanted to ask you about if you were going to look for a signature for life on another planet. I mean, now we have the big Webb telescope, what is it that you would look for? I mean, I agree the eukaryotic, getting to a eukaryotic state is going to take some time. There's a lot of, issues that are involved, but seems like life comes pretty quick in terms of prokaryotic metabolic.
Seems that way to me, yes.
Yeah.
I think I would look for chirality, because you can get some level of chirality from mineral catalysis or various physical processes. But as soon as you've got enzymes, you are more or less obliged to be doing hand in glove type chemistry. And when we talk about chirality, we often think about, well, all the amino acids are in the D form and all the sugars are in the L form and so on. And so it was one or the other. But the lipids are really interesting here, because the glycerol phosphate head groups are the opposing chirality in the bacteria and the archaea.
Yeah.
And so the three large groups of molecules that have this chirality, two of them are universal between bacteria and archaea, but one of them is actually showing, which to my mind says it's about enzymes. It's actually not about some strange physical process. It's just about, are you approaching it from above or from below as an enzyme? And then you'll. And so I think of chirality as a product of biology, and that means that if we see in any planetary atmosphere a very strong chirality, then I think that would be a sign of life. Because I don't know, would we be looking for NADH or ATP? I can't imagine that we would be. But and if you're looking for things like acetate or pyruvate, then that's something that a hydrothermal system can produce anyway. So it's really hard to know where you draw the line. And I think chirality is where I would draw it.
Do you know whether anybody's done any chirality measurements in these meteorites that have come from Mars or?
You know, I don't. I assume they have. I would be amazed if they hadn't. It's not something that I followed very closely. I'm sure if they had found that they were really chiral, I would have heard about it. So I'm guessing they're not, but yeah.
Let me pose a question that either of you may answer. This is from Patty, who says some bacteria that have an obligatory coexistence with a eukaryotic host have small genomes, minimalist metabolism, scavenge fatty acids from the hosts, and have membranes that contain eukaryotic lipids. Would you expect them to have a more complex cytoskeletal structure?
Interesting question. Jennifer, do you want to have a go first?
Well, if they have a small genome, probably not. But yeah, I mean, I'd like to know more about that bacteria in terms of its eukaryotic. When you say it has eukaryotic lipids, does it have sterols? I would argue that if it's not, based on the proposal that I made, which there's no evidence for, but it's just the way I like to think about it. If that bacteria doesn't have these complex lipids that can phase partition, you're not going to get any kind of complicated cytoskeleton. That's going to couple to that membrane and drive.
I suppose the question is, if it's stealing its lipids from the eukaryotic host cell, then effectively it's got stolen complex lipids. Would it then?
Yeah, no, that's a good point. Yeah, no, that's a very good point. Yeah.
I will go out on a limb and say I don't think it would, because I think the energetic constraints would be the problem for it there, that it effectively would have the potential to do these sophisticated things with its lipids. But I've thought for quite a long time about why you couldn't just use a bacterial plasmid, for example, instead of so I would see mitochondrial DNA as more or less like a plasmid sitting inside the mitochondria, and then you can think, well, you could take out the mitochondrial DNA and stick it next to the plasma membrane, and then scale up a bacterial cell to a eukaryotic size, and let's put tens of thousands of copies of this little mitochondrial genome right next to the membrane. Why is it not going to work? Because we never see that really.
Right.
Now, I think one of the reasons it doesn't work is that there's very little advantage for a bacterium simply being bigger for its own sake. It just takes longer to replicate itself then. So if you think, well, okay, if you make yourself twice as big and you're a streamlined shape and so on, then you can make twice as much ATP, and so you have an advantage. You've got more ATP, but you've just taken twice as long to replicate yourself, and so you're going to lose out to smaller bacteria. So the selection pressure on bacteria is pretty much always going to remain small. But the other reason I don't think it's going to work is that if you've got hundreds of copies of a genome and they're all contributing to a combined phenotype, which is, let's say, the membrane potential on the plasma membrane, even if it's invaginated, then selection can't see mutations in any one of those genomes because they're all contributing to a common phenotype. So you can accumulate a mutation here and a mutation there. They're not going to have changed the membrane potential much. So you're going to accumulate mutations slow until you have a mutational meltdown. Now, if you've got a single mitochondrion, and it's got, so this is something that kind of dawned on me a few years ago, that why do we have fusion into whole networks which you show so amazingly and beautifully, but you've got a kind of laminated network of all of these mitochondria. But if you block them fissioning again, then everything degenerates after some time. So why is fission, mitochondrial fission, so important? And it kind of just struck me that that's why just plasmids don't work, because what you have then is a genotype-phenotype relationship. You've got a mitochondrial genome inside a mitochondrion, where if you've got mutations in that genome, then it's not going to be able to maintain a good membrane potential, and it can be targeted by autophagy and broken down. And so by fissioning, you re-establish a relationship, a causal relationship between the genotype and the phenotype. You can select against things and dispose of the junk. Whereas if you've got a kind of common phenotype, then you can never do that. So I think there's several reasons why bacteria don't want to get that big.
Yeah. And there's a lot of reasons why you want to use small units for that type of mitochondrial evolution. I mean, the mitochondrial, I mean, we use mitochondrial DNA to track human evolution, human migration. And it's because of this rapid, ability to mutate, and on a much rapid scale than what we see in the nucleus.
Well, then there's selection as well in the germline. In the female germline, you have this combination of fast rates of mutation combined with selection for the ones that work. So you're not producing many children with mitochondrial diseases. It happens, right. but it's, given that it's mutating at 10 times faster than the rate of the nuclear genes, mitochondrial diseases are amazingly rare.
Mm-hmm. Mm-hmm.
Interesting. So Jennifer, we have an interesting question about plasmalogens, which in eukaryotic cells have both an ester and an ether linkage.
Yes. Mm-hmm.
So Joshua would like to know, do you have any ideas as to the emergence or the importance of plasmalogens in eukaryotic cells?
I know that peroxisomes are dealing with them, and some of these plasmalogens are involved in myelination of nerves. It might, these ether lipids could be very, make the membrane more resistant to, being permeable, permeable. I don't know, but it's very, very interesting. Yeah.
What do you think, Nick?
I don't have an opinion on that. I think I'm out of my depth there.
Okay.
But it's connected to myelination of nerves.
It's connected to what, sorry?
Myelination, nerve myelination.
Yeah. Right, okay.
So we have a fundamental question for both of you. Could each of you please define life?
Well, I started out by refusing to define life, and I actually think it's the wrong question, because life is a process. It's something happening over time, and so, is a virus alive? Most biologists would argue about whether a virus is alive or not because it doesn't have its own metabolism. I see a virus as basically as being alive because they're nasty bastards, and they take over the metabolism of the cells that they live in very sophisticated ways. They evolve, they change, they mutate, they do everything to do with life apart from metabolism, and they don't need that. But then, we are far more parasitic on our environment than plants are. We need to eat other things. So in some sense, like a virus, we are also parasitic on the environment. And then you think, well, plants are as well, in the sense that they still require CO2 to fix and water. They're not going to grow in a desert or in the dark. So, they require things from the environment to enable them to grow. And by that definition, it's not a definition, by that way of seeing it, a virus just requires more things from its environment to be able to grow. And so you could then think of life as being parasitic upon an environment. And by that, then what about retroelements? Are they alive? By that, they take even more from their environment than a virus does. And it's impossible to know where to draw the line. There isn't a line. There's just simply different forms of parasitism on an environment. And it's amazing if you see the world that way, just how beautiful it all turns out to be.
What do the I'm struggling for the word. I come up with officials, but what is the sort of organizational definition of life these days?
I ignore it deliberately, so
Okay.
I don't know.
We have an interesting question. If the proton, the motive force, is only about 200 millivolts, that isn't a lot. So
well, it is a lot if you think about the field strength, because we're dealing with a membrane which is 5-6 nanometers thick. And so the field strength is in the range of 30 million volts per meter, which is equivalent to a bolt of lightning. So it really is. Now, there's another interesting thing. I'd be interested to know what you think about this, Jennifer. There's a dirty secret in bioenergetics, which is that it's very, very difficult to measure the membrane potential. And it's usually done with fluorescent dyes, with lipophilic cations, which are supposed to, so the mitochondrial matrix or the inside of the cytosol bacteria is relatively negatively charged. So these cations will accumulate in there, and they should cross the membrane because they're lipophilic. And it all makes sense, but they interact with proteins in ways that are difficult to quantify. And the structure of mitochondria has become much more interesting, really. I used to think of the Christie structure as basically open and continuous with the intermembrane space, but they're not. The OPA1 complex closes the Christie. They're very narrow. They're very confined space. There's not much water in there, and it's not continuous with the intermembrane space. So that's how the lipophilic cations can get in,
Right.
Because they're going through the intermembrane space. They're never crossing something with a really strong positive charge, as they would if they crossed the Christie. But when people have tried to take measurements of the actual membrane potential using microelectrodes, they tend to measure 10, 20 millivolts, very, very low. Now, there are still people out there, including some very good scientists, who have still challenge the chemiosmotic hypothesis on that basis, that you measure it using the kind of neurological tools that people use for looking at neurons, and instead of 200 millivolts, you get 10, 20 millivolts. What I think they're measuring, though, is the difference between the cytosol and the matrix, not between the matrix and the Christie space. So then the question is, and this is my question to you, Jennifer, because I don't know how you do it. How can we measure? And it's hard to think of any other way than using dyes, but maybe a lipophilic anion or something that's going to accumulate in the Christie space and tell us what the membrane potential is between the Christie and the matrix. Now, the old measurements that Peter Mitchell and Jennifer Moyle used to do, looking at changes in pH, they estimated the membrane potential to be in the range of 230 millivolts. And that climbed down, and it's calibrated now using the lipophilic cations. And you won't read very often of a membrane potential of above about 180 millivolts. But I don't know what it's being calibrated with. People try and bacteria by the rotation speed of the bacterial flagellum, and it doesn't correlate very well at all. So I don't think we have a very reliable way of calibrating what the actual membrane potential of the Christie is using the lipophilic cations, because we don't have any other way of getting. Maybe we can do it by EM tomography and doing the measurements with the matrix values and then calculate the volume and calculate it that way. That might tell us. But this is a question for you as a cell biologist who knows all about ways of measuring these things, because I'm at a bit of a loss, but I think we're on the point of something really fundamental here.
That's really cool. I mean, so we do focused ion beam scanning electron microscopy, and recently have been introducing these dyes, these membrane potential dyes, into mitochondria. And you can then do correlative imaging. You can correlate, basically do high pressure freezing of the cell that's just had the dye added. And so it's cells frozen. Turns out the dye still is fluorescent, and and you can really look at it. You can look at it relative to the rest of the mitochondria. So it could be that that system could be used. And we haven't been thinking of it from the perspective of trying to get a more accurate measurement of the membrane potential, but maybe we should talk to you more about it. I'll send you an image of what this looks like. And yeah, I mean, what's interesting is that we see variability in the intensity of that membrane potential dye across different mitochondria.
Yes.
We see it
between Christie as well, I understand.
Yeah. Mm-hmm. Yeah. So
Jennifer, excuse me, but I was reminded that you didn't have an opportunity to provide your definition of life.
Well, I mean, I think I will agree with Nick that it's a very, I mean, I've been in many conversations, debates, arguments about the definition of life. And I think it's, you know, basically it's a process, it's energy, it's an energy flux. I mean, that's the typical definitions of, you know, when people talk about self-replicating, you know, containing DNA, you know, a membrane.
It's kind of more of a description than a definition though, isn't it?
Exactly. And I think what life is basically being able to harvest energy to drive processes in a way that's way out of equilibrium. But I realize that then allows you to extend the definition to something bigger potentially than cells. And like, you know, is the whole earth a living thing? You know, people talk about Gaia, you know, this whole idea that earth is it's a system, it's a living system. It's not made.
And it has a structure very similar to a cell in the sense that it's, if you like, reduced and relatively alkaline in effect inside and acidic and oxidized outside. So it's, there's a very direct parallel between the earth and the structure of cells.
Yeah. I'm
not saying it's alive, it's just that cells mimic the structure of the earth. There is a definition of life, a working definition of life that NASA have, and I probably can't get this quite right, but it's something like a self-sustained Darwinian system capable of evolution or something along those lines. And I rebel against the word self-sustained because it denigrates the environment to the point of non-existence. I know what they mean, but it certainly doesn't emphasize an environmental disequilibrium is driving everything. And the other, you know, people can poke fun at it as well and say, by that definition, only, you know, a pair of rabbits is alive. A single rabbit is not alive because it can't actually copy itself. So, you know, all of these definitions have holes in them, which are almost risible holes. And you're left with this feeling, well, are we actually learning anything from making fun at other people's definitions? I mean, I think there's a problem with definitions altogether when it comes to life, just because it's a continuum.
Well, on that sober note, I have to note that we have reached the limit of our time.
However, gosh, that was fast.
However,
awesome.
On behalf of the many, 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 many people who watched and participated in this, I just want to be the spokesman to thank you both for informing, exciting, and certainly stimulating the hell out of our thinking. And it makes me realize that there are probably at least 5 other subjects related to the combined interests of you two that maybe we can approach on another occasion. So thank you very, very much for taking part in this, and we gratefully appreciate it. I'm sorry that all good things have to come to an end.
Thank you very much. It's been wonderful.
Thank you so much.
Thanks, Nick.